Hello everyone. So this will be our next discussion for the topic called heredity. So what we have right here in this session, so we will try to find out how DNA makes protein. So in this case, we have already discussed what is DNA all about and its counterpart, which is the RNA in the first discussion. So we already know its functions and parts. Yeah. So in this session, we will going to discuss how DNA makes protein. So what do you have right here? Excuse me. So what do we have right here in our slide? So in this slide, you will see Morse code. So in every dash that you see here, all right. So in every dash that you see here, yeah. So let's go back again. So in every dash that you see here, it corresponds a certain letter. So for example, we have this D over here. So we have dash and two dots. So in physics, we knew that the Morse code is a product of electromagnetism. So along press of your telegraph means dash, and short press of your telegraph means dots. Yan. So combination of dots and dashes creates certain letters. So you need an encoder. You need someone who can read this, who's expert enough to read this code that we have right here. So what is this code all about, and what is this code has something to do with our topic today. So the Morse code, yeah. So it is invented by Samuel Morse. Alright, so there are other things that we have right here. So the numbers, yan. So then we have alright, so these are some examples of the scriptures found in the Dead Sea. So we call it the Dead Sea Scrolls, and I think it's the New Testament of the Bible. Yan. So you need a translator. You need an expert who can translate these words into modern language that we all knew. So it's either in English or other languages that you want. No, that's the reason why your Bible is translated into different languages. Yan. So this is the Dead Sea Scroll. So what is this one? Has something to do with our discussion today. So as you can see, you cannot read this one without someone who knows how to translate. So someone who knows how to translate. So that particular phrase has something to do with our discussion today. So let's go. So the title of our session today, or our topic, is how DNA makes protein. So in short, we have protein synthesis. Yan. Okay, so as we all know, the function of DNA is to make this is the blueprint of life. So anything around you, animals, plants, contains DNA, and the way they look like is within their DNA. Yan. So how does DNA makes protein? So let's start first with the first thing the DNA does is DNA replication. So DNA replication. So we have this DNA replication happens during mitosis. Yeah. So mitosis. Yeah. Yeah. So it happens during mitosis. So DNA replication, so that all cells must have an must look like, all daughter cells must be look like, or must be look alike with its parent cell. So that's mitosis. Yeah. So one of the things that happens during mitosis is DNA replication. So let's discover how is the replication possible. So What is DNA replication? So this is the process by which DNA makes a copy of itself during cell division. So technically, as I said earlier, so we have here mitosis. Yan. So it happens during S phase of cell division. So we have your S, G1 phase, yan. So M phase, yan. So G2, yan. So it happens, this replication thing of your DNA happens on S phase. So we call it synthesis phase, yan. So next. So in this picture, this is in summary, so the DNA replication looks like this. Yan. So it's just uncoiling itself and creating new strand out of the parent strand that we have right here. So this is your parent strand, and this is the new strand that is created, right? So in this side we have this parent strand, and on this side, so this is the new strand. So basically it looks like so the new strand must follow also the complementary rule, base pairing rule. So A to T, C to G. Yan. So that's your DNA replication. But technically, so what do you have right here? These are some ideas on how DNA literally replicate. So we do have semi-conservative model and conservative model. So another one, we have dispersive model. So alright, so let's start with the semi-conservative model. So how does DNA divides in a semi-conservative model? So in parental DNA, it will divide. So it will uncoil, then alright, so as you can see, uncoiling of the parental DNA will produce, right? So will produce a new strand of your DNA. That's the blue one over here. Yan. So it started with the pink one, then after replication, so it will produce the blue one over here. So that's the new strand. And then after second replication, so it will look like this. Yeah. So it will look like this, the one at the bottom. So as you can see, did the pink one disappear? No. So it does divide, and then what happened is new strands did multiply. So that's what happened here. Next one. So in the conservative model, so we'll start with the parental. What happens here is that the parent, this one, the pink DNA over here, uncoil, then it produces this blue one over here, and another pink DNA over here. This is after first replication cycle. So just like the other one, after the second replication, this blue one over here produces obviously blue new strands, and the other one, so the reason why it's called semi-conservative, so it is retained. So whatever characteristics does this parent DNA have, it is maintained until to the second replication cycle. So as you can see, the way the second replication looks like is the same with its parent DNA. So in the semi-conservative model, so the pink strand of DNA, this one, so the pink strand of DNA is separated. And why semi-conservative? It is still maintained, but not totally the same with the parental DNA. So that's how semi-conservative model. works. Yan. And in the dispersive model, it's a bit complicated. So, in this model, the parental still in pink, but what happened is it's just like the pink parental DNA happened to be chopped into several pieces, as you can see here. And so it is chopped into several pieces. Then the blue strand is produced in between the pink strand that we have right here. So as you can see, the reason why it is called dispersive, because the pink strand, from the word itself, dispersive or scattered, yan. So that's the appropriate word. So scattered on the second replication cycle. So it is scattered on the daughter cells. Yan. So this is now the new strand of your DNA. So what do you think? So these are some three ideas that is related to how our DNA replicates. So as we progress, you will know which of these ideas are said to be happens during DNA replication. So will it be semi-conservative? So will it be semi-conservative? Will it be conservative model, or will it be dispersive model? So let's try to find out. So in this case, so how DNA replicates? So alright, so as you can see, this dots over here, this is the origins of replication, and this is the point where the parent DNA will start to create its daughter strand. So this blue one that we have right here, this is the daughter strand. So starting on that origin, so starting on that point, so this blue strand will start replicating. So the direction of the replication starts on both sides. And so as it elongates on the third picture below, so as it elongates, it will connect eventually. And this side will connect eventually, and this side will connect eventually. So that's how the replication fork direction goes to. And after the replication, we have now your two daughter DNA molecules. So one over here, and one over there. And there we go. So that is after the replication. So the question is, how is it possible? So what are the things in this process that makes this replication possible? So the replication is possible because there are some enzymes. There are some enzymes that are included in this replication process. So we have your helicase and we have your polymerase. Alright, so there you go. So let's start first with the helicase. So in order for your DNA to be separated into its single strand coming from the double helix strand, so as you can see, it is in the double helix over here. So in order to separate that one, so you need an enzyme called helicase. Alright, so remember, if you imagine a zipper, so a zipper will not open if you will not pull down the one that is on the middle, the one that opens the zipper. Yan. So that's how this helicase look like. So yan. So just like a zipper, this is the one found in the middle. Yan, this helicase. And this helicase cuts open, or it separates the two-stranded DNA into single strand that we have right here. Yan. So at this point, yeah. So it separates the double-stranded DNA into single strand. Right, so that's the helicase. Then we have your DNA polymerase. The DNA polymerase is the one that creates the daughter strand that we have right here. Yan. Okay, so as the DNA creates new strands of DNA, it must follow the complementary base pairing rule. Again, A to T, C to G. Yan. So, there you go. So, that's your DNA polymerase. That's its work. Yeah. So, as you can see, these are your nucleotides, and then eventually, these nucleotides will become part of the new DNA after replication. So, oh, there's another DNA polymerase over here. Yan. So, its work is the same thing, the one on top. Okay. So, that's how DNA replication works. Okay. Alright, so if you look at the chromosome, during replication, so technically your chromosomes looks like this. There you go. Yeah. Okay. So it looks like this, and it has one chromatid. Now after replication, it will have two chromatids. Alright, so yeah. So after my mitosis, so the cell divides, then again the two chromatids will become one chromatid again. So there you go, right here. So this one happened because of mitosis. So remember the process in mitosis called anaphase. Yan. So in the anaphase, it will totally separate this chromatid that we have right here. From two chromatids, it will become one chromatid. So after telophase, it will look like this. So we have prophase, metaphase, anaphase, and then telophase. So this is possible. So this is seen during mitosis. Yeah. Yeah. So that is how DNA in chromosome, which is located in the chromosome, looks like after mitosis. Yeah. And as I said earlier, during mitosis as well, replication of DNA happens. Yeah. So what's the goal of DNA replication? One thing is to ensure that the somatic cells in a multicellular organism carry the same genetic information. So that's very important. You will look awkward if you have a skin which has different color every time it divides. Yan. So that's technically the goal of your DNA replication, and one of its minor goal under this umbrella, so under this goal, is maintenance. Alright, so maintenance. Yan. So maintenance and growth. Yeah. Alright, so that's technically the goal of DNA replication. Okay. Next one. Alright, so next up, how are proteins made? So the one that creates the blueprint of our proteins, or the blueprint of our body, is in the DNA. So as we discussed the DNA last time, that DNA functions as the blueprint of life. So the proteins, the pattern on how proteins are created is within the DNA. So how are this pattern be able to be decoded? Yan. So in order to make these particular proteins. So alright, so what's the main idea here? So the DNA genotype is expressed as proteins, which is the molecular basis of phenotypic traits. So remember when you were grade 8, those phenotypic traits, so we have dominant, then we have the recessive thing that we have right there. So these genes that we have right here technically are proteins. Technically are proteins. So if genes are made up of proteins, so DNA is the one that creates proteins. Okay? So in this process, there are two stages. So we have transcription and translation process. So very straightforward, transcription and translation. Genetic information written in codons is translated into amino acid and sequence. By the way, what is this codon all about? So Codons are 3-letter nucleotide combinations. Yan. For example, we have A, T, G. Yan. There you go. So, codons are 3-letter combination. It must not be less than 3 or greater than 3. So, codons. Yeah, that's very important. So, let's move on. Alright, so in protein synthesis, we will need the use of this codon chart that we have right here. So later we will discover how to use this one. And as you can see, this codon chart contains amino acid. There you go. So these are amino acid. So we have phenylalanine, leucine, isoleucine, methionine, so on and so forth. So later we will discover how to use this one. And also this amino acid can be unlocked using 3-letter codons. Yan. So these are called codons. Yan. Alright, so for now we will just have a glimpse of it, and we will take a look. We will use this one at the last part of this discussion. Alright, so just like the Rosetta Stone that is found in Egypt, yan, this was written by Egyptians 1000 BC ago. Yan. So this Rosetta Stone cannot be if you read this Rosetta Stone, even me cannot understand what is written on it. But if I hire someone who is an expert in Egyptology, he or she may be able to read this one. So, in short, you need a translator. Yan. So, you need a translator. So, how to do that? So, how is this one related to our protein synthesis? So, let's take a look. So, transcription. Transcription is the process of creating an equivalent RNA copy of a DNA sequence. Yan. It creates mRNA. So, it creates the mRNA. So, in transcription, one thing that you need is this one. Yeah. So, one thing that you need here is the mRNA. Yeah. So, what's the process? Equivalent RNA of the DNA sequence. Yeah. So, let's have a short recall of what is complementary base pairing. In DNA, we have A, T. Then we have C, G. Yeah. In RNA, we have A, U. So, there's no more T on RNA. And on C, G still. Yeah. So, now, let's take a look how transcription process happens. Alright, so here we go. So here we go. So we in the transcription process, we have an RNA polymerase. So when you hear the word polymerase, it's something that creates new strand. Alright, so same as your DNA polymerase. So then your RNA polymerase will approach this this DNA strand that we have right here. So it will start first on the side of the promoter DNA. So from the word itself, promoter, it promotes transcription. Yan, it promotes protein synthesis. Yan. So this is the start, and from the word itself, terminator. Yan. So this will end up the transcription process. So as the polymerase, as the RNA polymerase goes into the gene itself, yan. So that's the initiation process, and the elongation process as it travels down the stretch of your genes. As you can see, there is a growing RNA that we have right here. Yan. So this RNA that we have right here is the mRNA. Yan. So what do you think? Alright, so what do you think? Is this RNA has the same sequence of the DNA gene that we have right here, or not? So leave your comments below. Alright, so we have the termination. Obviously, the RNA polymerase do not have all the time to copy everything on the DNA. So what it needs, what does is shown, what is shown here is the RNA polymerase only copies the strand of the DNA needed. Yan. What is needed only. Right? Not all stretch, or not all the DNA genes, or not all that long stretch of DNA gene must be copied. So only that is needed by the cell. So after the process of transcription, there you go, we have now your mRNA. Okay, so this mRNA now, so this transcription thing happens inside the nucleus. Right, so take note that one, that the transcription process happens on the nucleus. Alright, so then this mRNA over here will goes out the nucleus via the nuclear pore. Then the next process of protein synthesis will happen on rather outside the nucleus. So remember, transcription happens in the nucleus. Okay. Next, other picture or other diagram for the transcription process. So there you go. This is your RNA polymerase. And as the RNA polymerase zips down the stretch of DNA, it creates your mRNA strands over here. So as you can see, if this strand is copied by the RNA polymerase, it should have similar base on the, or rather it must have a complementary base on your mRNA. So if that is G on the mRNA strand, it will become C. Then on the DNA, if the DNA has A, and on the mRNA it must have U. So since T is not available on RNA. Yeah. So that is transcription. Alright, so the next process is translation. So the translation is the first stage of protein biosynthesis. Alright, so protein is not created on transcription. So the process there in transcription, you just only copy the DNA strand and creates an equivalent mRNA strand similar to your classmate who is copying your homework. Yan. So he's the one who make copy of your homework exactly word for word on your homework. And so he's your classmate who did the transcription. Yeah. So the production of proteins by decoding mRNA produced in transcription. So technically, no translation will happen if there's no transcription. So the first stage or the first process is very important. Next one, it occurs in the cytoplasm. So the other one occurs in the nucleus. Then this one occurs in cytoplasm. So why cytoplasm? Because ribosomes are located in cytoplasm. That's how. That's technically it is. And what are the things that we have right here? So we have tRNA, and it requires tRNA for interpreting the mRNA. So just like what I've said earlier, so the Rosetta Stone will not be understandable to everyone, or you cannot understand what is on the Rosetta Stone if there will be no person who will interpret what is written on it. So will translate what is written on it. So this is the Rosetta Stone. And this is your interpreter. The tRNA, the interpreter. Alright, so let's move on. As you can see, your tRNA over here. Yan. So your tRNA over here carries already the amino acid that we have right here. Yan. Carries the amino acid, and the tRNA carries the codon, this codon is what they called, this codon that we have right here are termed as anticodon. Yan. Right. So why anticodon? These are the codons coming from the mRNA, and the one that is carried by the tRNA is called anticodon. Why tRNA? Because from the words, from its definition, from its word transfer RNA, which means it carries the amino acid, yan, over it. So once this anticodon attach or being complement to the codon carried by your mRNA, yan, the amino acid that it carries correspond to the mRNA sequence. Take note, this amino acid that we have right here corresponds to the mRNA sequence, not on this anticodon sequence. Take note that one. So for example, we have your mRNA over here, which is the codon UUC, and the equivalent amino acid for that is phenylalanine. Alright, so take note that it's the mRNA. Yan. So the anticodon for AAA is, the anticodon for this codon is UUU. Yan. And AAA has an equivalent amino acid called lysine. Alright, so later you will know how it's done. So next one. So there you go. Alright, so the AUG that we have right here, if we look at the codon chart, it means start process. Yan, it means start. Literally, this will start up the protein synthesis. So start. Yan. And it carries the amino acid methionine. It means start. Right? So if there are start codon, there is a stop codon. So as you can see on the codon chart, there are codons that signals the stopping of process of protein synthesis. Yeah. Okay. Next one. All right. So these are the step-by-step process on how technically the protein synthesis or the translation works. So let's start with the first one, codon recognition. So the codon recognition is from the mRNA. So this pink one is your mRNA. RNA. Yeah. Alright. So your anticodon is carried by your tRNA. And it has the amino acid that corresponds to the mRNA. So as you can see, alright, so what is this black object over here? This one. So this is your rRNA. Yan. Right, so this is your ribosomal RNA. Okay, so or the ribosome itself. Yeah. So it helps the process of protein synthesis. Yeah. So the first step is the codon recognition. Then the anticodon will attach to the codon, and as it attach, it will create a link of amino acid. So this is an amino acid. Now once they are linked together, they are not called amino acid anymore. They are called polypeptides. Yeah. Or in short, we called it proteins already. So once your amino acid forms a chain already, so they are now proteins. So as it grows or as it elongates, right, so the one that is the anticodon that is not needed anymore in the process is ejected. Then the new anticodon enters. Here you go. So the new anticodon enters, then it will form another amino acid, and then it will form another peptide bond with that amino acid. Then it goes on this process again. Yan. It continues until you reach the stop codon. Then when you reach the stop codon, literally it will stop the entire process. So it will stop making the amino acid in the process. So the anticodon always adds up, like the one that we see here. So it always adds up, then it is ejected after it's done, then until you reach the stop codon. That's where the process will totally stop. Yeah. Okay. So next part. Alright, so what's the difference between transcription and translation? I made a Venn diagram over here. So transcription, product of transcription is mRNA. So transcription occurs in the nucleus, and transcription requires RNA polymerase. Whereas in translation, alright, so the product here in the translation is the polypeptide chain. Right? So on the other hand, mRNA, this is polypeptide chain, and translation occurs in ribosomes. Right? So translation and transcription occurs in nucleus. Translation uses various reagents to create the polypeptide chain. So what are those reagents? So we call it lang reagents. Yeah. Alright, so those reagents are very complicated. So yeah, so they use reagents to create a polypeptide chain. That's your translation. Now, what's the similarity? What's the similarity of those of both processes? So, alright, so both use cytosine, guanine, and adenine. Right? So, coming from the DNA, and all including uracil. Yan. So, as nucleotide that they use. And one thing is important here, no transcription and no translation will happen if there is no template. This is very important. Alright, so if you will copy the homework of your classmate, alright, so if you will copy the homework of your classmate, so if there's none, what will you copy? Yan. So you need a template. So that's how it's work. How it works. Yan. Alright, so you need a template. So there you go. That's the differences and similarities between transcription and translation. Next up. Alright, so this is the overall, or this is the summary of the process of transcription and translation. So the transcription happens in the nucleus, and translation happens on the ribosome. So we have your ribosomal unit over here. We have your anticodons over there. This is your codon. Don't forget, it will always start. Yan. It will always start, and there is always an end to the process. All right. So in this process, take note, everyone, that in this process requires ATP. Yan. So protein synthesis will not happen if there is no ATP and energy. And so how will you create new skin? How will you create new skin cells? How will you create skin color? How will you create a new hair strand? Yan, if you don't have the energy to power that process. So technically, yan, that's how it's done. So that's your transcription and translation. Right. So by the way, this whole process that we have right here, is under the process called protein synthesis. So I will not discuss anymore what protein synthesis is. It's very straightforward. Yeah, you already know what does what each word means. Yeah. So protein synthesis. Alright, so let's have a short exercise on how photosynthesis happen. So, alright, so there you go. So first up, so we have this DNA strand over here. And as we all know, we will always start with a codon. So always group them into three letter group. Yan, called codons. So yan, codons, we call it codons. Alright, so let's start first with cDNA strands, or complementary DNA strand. So during replication process, you only make complementary strand. So alright, so what is the complementary strand of this DNA that we have right here? So obviously A will become T, your T is to A, then G is to C rather, then G is to C, then A, then we have G. There you go. Alright, so this is the replication process. Replication. Yeah. And then DNA replication. So we will use another color for the other one. So in the mRNA, so RNA polymerase is used in this process. The process that happens here is what they call your transcription. Transcription. Alright, so we will have the pattern over here. Yan, so don't be confused. So we will look here. So transcription. So we have so we have U, we have A, we have G, okay. Sorry. Right, so we have C, we have C, we have A, we have G. Yeah. So there you go. So that's your mRNA strand. Now let's take a look at the tRNA strand, or this is the anticodon, be exact. And this process, this 2 here is your translation. Remember, translation happens in the ribosome. Right, so let's use different colored ink over here. So we have we'll use blue. Alright, so in tRNA, so let's just copy the opposite of this one. So we have U is to A, so we have A is to U. Yeah. We have G, we have G, we have T, and sorry for the writing. Alright, so we have C, and there you go. So AUG, GTC. So that's your tRNA strand. Always remember to group them into 3. Right? So we called it codon. Now, our problem is the amino acid. What is the amino acid? What is the amino acid equivalent? And where we will look for the amino acid equivalent. So take note, the amino acid equivalent, you should look on the mRNA strand, not on the tRNA strand. Take note that one. Alright, so let's use another color over here. So in this case, we will use black. Alright, so what is the amino acid? Oh no, don't worry if you don't have an idea what is the amino acid. So let's go back and take a look at the codon chart. There you go. This is the codon chart. So this is where we will look our amino acid. So let's go back to our problem. Alright, so let's take a look at the mRNA. So in the mRNA, we have UAC. Alright, so UAC. Alright, so the first letter is U. The U. The second letter is A. There you go. And the third letter is C. U, A, C. Yeah. So this is the third letter, UAC. So that is tyrosine. So we called it TYR. So just put the 3-letter word there, 3-letter abbreviation for that amino acid. So we have TYR or tyrosine. Alright, so next one. Let's take a look at the mRNA. Alright, CAG. Right, what's the amino acid equivalent of mRNA? CAG. Yan. So let's take a look. CAG. Alright, so first letter is C. Look at the second letter, A. G. There you go. So we have this one. Alright, so we have glutamine or LN. Yan. So, let's write this one down. So, the amino acid of CAG is so the amino acid equivalent of CAG is GLN or glutamine. Yan. There you go. So, that's right. So, that's the first exercise. So, let's have more exercises here about protein synthesis. So you can pause this video if you want to, so for you to try out for yourself what will be the answer on the blanks right here. So let's take a look at the complementary strand DNA. So we have G. So we'll use so we'll use a different colored pen. Alright, so we have G, we have C, we have T, we have T, we have A, and we have C. Yan. So that is the cDNA strand. So let's use another color. So let's use green for the mRNA. So the mRNA, G, C, instead of T, we have U, then we have U, then we have A, and we have C. There you go. So let's use another color again. Let's use blue. Right. So in tRNA, so we have C, G, A. We have A over here. We have U. Okay. Then we have G. There you go. So that's your tRNA. Then for the amino acid equivalent, okay, let's use black ink over here. So the amino acid equivalent of mRNA. So mRNA amino acid equivalent. So let's take a look at the codon chart. Alright, so I forgot to memorize the GCU. So GCU. Yan, there you go. So GCU. So first letter G, C, on this lane, C, U. So GCU is alanine. Yan. So let's write this amino acid there. So we have A, L, A. Alright, so next one, UAC. So let's take a look. Codon chart. Boom. So U, A, C. So that is tyrosine. So let's write this one down. Tyrosine. T, Y, R. So, tyrosine. So that's our second practice problem. Next one. Alright. So again, feel free to stop this video to try this one out for yourself. So again, so we will use another color, the red one. Alright, so we have the T to A, we have T, we have G, we have T, we have A, and we have A. Yeah, there you go. So that's the complementary strand. Next one. So we have green one for the mRNA. So in the mRNA, so we have alright, so we just copy this one. So we have A, we have U, we have G, we have U, we have A, and we have A. So there you go. So for the tRNA, we will use dark blue, blue rather. So we have T, or rather, sorry. There's no T in RNA. So we have U, we have A, we have C, we have A, and we have U. Alright, so that's the tRNA or the anticodons. Alright, so again, the amino acid. Alright, so the amino acid, we will get the codon pattern of your amino acid coming from the mRNA. So let's start with the AUG. So AUG, so let's take a look at the codon chart. AUG. Alright, so A, this is the first one. U on this row, and G. So AUG is methionine, or literally start. So let's put it there. So we have start. There you go, start. You can put start or methionine if you want to. So if you can also use the abbreviation MET, yeah, or your start. Next one, so we have UAA. So UAA. So let's go back again to the codon chart. So we have UAA. So fortunately, we have the stop codon over here. So we will write it there. Alright, so we will write it there. So we have stop. Literally, you just write it. Write stop. What is written on the, you just write here what is written on the codon chart. If it is said stops, then write it here stop. Yeah. So basically, this is how your replication, translation, transcription process. Yeah. So the protein synthesis does transcription and translation. Yeah. So alright, so on the last part of our video that we have right here, so will you please take note that, alright, so this process that you have seen over here, so this is the central, alright, so we called those process that you have encountered earlier, we called it the central dogma. While central dogma, this is some in short for pattern, universal pattern. Alright, so universal pattern, or all organism does this particular pattern, which is under the protein synthesis, we have the transcription and translation process. So in this diagram, you will see the DNA is being transcribed by the RNA, and the RNA is being translated to become a protein. So there you go. So the RNA can reverse transcript, yan. So it can perform reverse transcription, and don't forget to have your replication. So yeah, so the protein synthesis happens as the organism grow, yan. So if you want to grow more new body part, new body part. So if you want your body to grow, yan, so or rather than in maintenance, yan, in repair of body parts, so yan, protein synthesis is very important, yan. So production of melanin, production of enzymes, yan, that is protein synthesis is very important on those processes. Right? So yan, there in this diagram as well, it includes some processes that happens only on the laboratory, yan. So these are special processes. So a DNA can become a protein immediately without going to without doing protein synthesis, and this is applicable, or this available only, or happens only in the laboratory. So you can synthesize protein out of the DNA, yan. So if you heard some researches that some DNA produces protein like the one insulin taken from a particular animal and creates insulin. So this is that process, yeah, the one below here. All right, so take note, the central dogma starts with transcription, translation process. So This is your central dogma or the universal pattern for photosynthesis. All right. So basically, that's the end of this session, and I hope you did understand all the things that we have discussed today. So have a good day, everyone, and God bless.